And this, if you really see IL-12, how it looks like when you put like more focus on it, it is heterodimeric cytokine. When I say heterodimeric cytokine, that means it is having two subunit. And when I say hetero, that means these subunits are different. When I say homodimer, the two subunits which are similar or same molecular weight. So this is essentially made up of a protein. You have two subunits. One is P35. The second subunit is called P40. P40 and P35 is nothing. It's a molecular weight of the protein. When you run this cytokine on the SDS page gel, right? You know SDS page gel. So if you put the protein on the SDS page, it can run based on the size, right? From positive to negative, like that. So it comes at P35 is kilodalton, and this is a P40 kilodalton. And when it's actually made within the cell, it made as a P70. So if you combine these two things and run on the gel, it comes P70 at 70 kilodalton. But you will say P40 and P35 is equal to P75. 5 kilodalton is very difficult to resolve. So P70 is approximately, people call it, right? The complexity is not only this level, the complexity is second level. As we try to go deeper and deeper, things get complex. So therefore, we should always, whenever you go anything more deeper, things are not as good, right? Things are good from the outside, but things are a bit different when you see them too close, right? So that's what it happens here. And when I see this receptor of IL-12, is even more problematic in the sense, I'll tell you why it's problematic, not now, maybe in a couple of slides or maybe more slides. The receptor of this cytokine is also heterodimer, right? It has two subunits. One is called IL-12 receptor beta 1, IL-12 receptor beta 2. It's just about the nomenclature. Beta 1 was discovered at the first, right? It may be having beta sheet, beta 1, beta 2, right? So this heterodimer make this functional complex called IL-12 receptor, and this IL-12 receptor, functional IL-12 receptor, express on the T cells. So just imagine this on the surface of the T cells. Just imagine this is being produced by the dendritic cells. And when the T cells are getting activated by two signal, and then the third signal is this cytokine, it activates this transcription program called STAT4. Each of these different T helper cells is having different stat. STAT4 for Th1, STAT6 for Th2, and STAT3 for Th17. And then STAT4 actually induces Tbet, which I told you, and induces more like a Th1 cells. How we prove it? Because this looks like I draw it myself, but no, we prove it by this experiment. And this experiment is nothing. We created these knockout mice, not me, but the people who did these experiments at that time. So if you create IL-35, P35 knockout, means the P35 is deleted in the mice, or you create P40 knockout, either of it, what you will see? You will expect that, if you remove this or this, you expect this whole pathway to collapse, right? And how you prove it? You take these mice and you compare this a wild type mice where everything is intact, everything is normal. And you compare the knockout mice with the wild type mice, and you just infect these mice with a mycobacterium, which is a pathogen, intracellular pathogen. An intracellular pathogen is cleared by Th1 cells. So in this case, there are no Th1 cells. Then what will happen? Bacteria will grow, because you are not able to kill the bacteria because the whole pathway is not there. And as a result, these mice are susceptible to mycobacterium infection and eventually die because of the infection. Same way they created this knockout, either this chain knockout or this chain knockout looks like same. Mice dies, no Th1 response. If you create STAT4, same. So whole pathway, that means this is required, this is required, this is required for functional Th1 cells that eliminate intracellular pathogen. I told you that when the Th1 cells produce interferon gamma goes in autocrine manner to further amplify Th1 cells. So people also prove it, this pathway, interferon gamma through the receptor, activate this JAK1, STAT1, Th1, further amplify Th1. And if you make either of the knockout in these, either this or STAT1, no response, again susceptible to the infection, dies. So now you connect the dot from IL-12 to this to IFN gamma receptor and say this whole pathway is required for Th1 response. If you remove Th1 response, you are more susceptible for this. So these things can be done in the mouse. It cannot be done in the human because we are not the mouse, but we'll like to learn from the mouse. So in that case, what people do, there are individuals in these pathways are mutated. Those individuals are much lesser, but these individuals in the Th1 pathways are mutated. They are susceptible to infection, right? So therefore, if they get infection, they cannot clear and they have require higher dose of antibiotic and all those kind of things. What I'm trying to tell that what you actually learn in mouse immunology is really actually translate to the human in a way, and then you can design the drugs by on these pathway to either enhance these pathway or decrease these pathways the way whatever you want to do it, because immunology or immune system is a double-edged sword. If you decrease infection, you increase autoimmunity, right? The America at some point of time was there a lot of infection was there because they were also not developed country, they were developing country like us, and that time they have more infection, less autoimmune disease. Now they have lesser infection because they are more clean, more thing, but they're having more autoimmune disease. So India now infection is there, but slowly we are getting down and we are rising the autoimmune disease, right? So you can play the immune system the way you want and to create new therapeutic things. So now I put together the Th1 pathway, and I'll then tell you the story of how the next transcription factor was discovered in this pathway. So this this both goes and activate or transactivate this IFN gamma gene locus. And once this IFN gamma gene locus is open, it make IFN gamma RNA, mRNA, and that mRNA is converted into protein, and that protein is secreted out, and that's the function, right? So people wanted to understand what is the really a transcription factor which actually binds to IFN gamma locus or promoter. And this is the paper got published in Cell in 2000, where they're trying to identify the transcription factor, so-called we call master transcription factor, which does only this, nothing else, right? So they actually generated the Th1 and Th2 clone, Th1 and Th2, and then they did a lot of screening of different transcription factor, and one of the transcription factor they identified is called TBET. Is a protein name, and TBX21 is the gene name. So you can see that TBET is actually expressed only in the Th1 cells, not in the Th2, whether it's a day 1, day 2, day 3, day 5, day 8. Everywhere is the Th1 cells are expressing, not the Th2. That indicate that this particular protein is very selectively and specifically is being expressed by the Th1 cells, but not on the Th2 cells. Then they further went on to this. This is a big paper. I try to take something out of it, and then they try to clone this protein TBET, and they did this luciferase assay. What is luciferase assay? Luciferase assay where you want to see a promoter activity. Let's say I want to see IFN gamma promoter activity, because promoter of a gene is something by which your gene will be transactivated or transcribed. If you don't have a promoter, right, you cannot transcribe it. So therefore, people clone the promoter reason, which is normally what happens, the proximal, the start site of the gene, you take minus 2 kb. So that become your proximal promoter, 2 kb from the start site, right? And then in this 2 kb, you fuse it with the luciferase gene. So luciferase gene is just a marker by which you can see through the machine. Luciferase luminometer, we call it luminometer. It's a like firefly, you know, Jugnu, right? This is what they have cloned from this Jugnu, and this is called firefly. And then you have this gene promoter fused with this luciferase, and then you put Tbet into it. So since it is IFN gamma promoter, you have luciferase gene, and we are assuming that Tbet will having a binding site on the IFN gamma promoter. If it is bind, you have luciferase. If you don't bind, you don't get luciferase activity. So that's what they have done here. Wherever they have IFN gamma luciferase genes put together with the Tbet, you see this nice expression of the black one. But if that is not happening with the IL-4 promoter, IL-4 is a Th2 cytokine. So Tbet is not doing anything, is as good as control. And they have taken IL-2 promoter, which is also T cell cytokine, a growth factor. So Tbet is not able to transactivate. So it becomes very specific for IFN gamma. And then they have done this experiment where they have overexpressed Tbet. Overexpression means you try to do an force experiment, or you what you call it exogenous. You clone the Tbet in a retrovirus kind of setting, and then you put these retrovirus on the T cells. The virus will infect the T cells, and then this whole Tbet will go inside. You are putting more Tbet. So wherever you put more Tbet as compared to vector alone control, you have in 24 hour more IFN gamma, 48 hour is further more, and 72 hour at different time point you see more IFN gamma. So taken together, this actually become a transcription factor which now is important for interferon gamma positive Th1 cells. So now what you learn from this whole thing, IL-12 IFN gamma important for this pathway, transcription factor STAT1 we talked about, and Tbet. So you may say, why is so important for us? ये तो हो रहा है, सेल के अंदर हो रहा है, हमें इससे क्या करना, right? What you can also learn from it, that you identify some of these key things, key pathway. Many multiple things are happening which we cannot really pinpoint. But some of the key point which we pinpoint, we can design a molecule, we can design inhibitors, we can design activators, right? And now when we design it, then you can manipulate this pathway depending on your need. So that is why the basic science of fundamental discovery can lead to generating new therapeutics. This is the one thing. So now type 1 immunity, which is why I'm saying this pathway is important to know. Some of you know, maybe knowing about CAR T cell. What is CAR T cell? Chimeric antigen receptor. Oh, I thought Mercedes you are saying, right? CAR T cell is one of the hot thing which is happening. I'll not really focus here. So this is like type 1 immunity potential for cancer. Now you know the pathway. Th1 cells are really important for tumor immunity. If you have tumor, you want to promote more Th1 cells, right? So how you can promote? You can promote by upregulation of Tbet, because Tbet, what it does, make more interferon gamma. You can promote the cytokine which makes Th1 cells, IL-12 one of it, IL-18, these are the two cytokines which promote Th1 cells. If you put, you have more these things, and you can have other cytokine, other pathway. So what we can do, you actually take antigen. Let's say, I'll give you an example of CD19 CAR T cell, what we are talking about. I think many people must be knowing that CD19 CAR T cell is good for eradicating lymphoma, B cell lymphoma, right? And B cell lymphoma is something where your B cells actually become malignant and they keep generating a lot more, and as a result you die. So now you actually make an antigen which actually bind to B cell, and we call it SCFB. SCFB is a single chain fragment variable region of antibody. So when you say antibody, antibodies are like this, like Y shape. you have two light chain, right? In the light chain, you have the variable region and the constant region. You have two heavy chain, variable region and the constant region. So what we try to do as CFB, we take the variable region of the heavy chain and the variable region of the light chain. So you have like this. So this is your heavy chain, variable region, constant region. And this is like this. And you have the light chain, which is only this much, variable region and the constant region. So what you do, you take the variable region of heavy chain and the variable region of light chain. Why we are taking variable region? Because these are the regions which bind to the antigen, right? We call them in the antibody, we call them paratope, which bind to epitope. Epitope is on the antigen. So paratope on the antibody. So paratope bind to epitope, and that's how the recognition happens, antibody antigen interaction. So what we are doing as a genetic engineer, and this is what we do very often in our lab, I think on daily basis, we are making a lot of new CAR T cells. So what we have done, we have taken this and link the variable region of heavy chain and the light chain through this linker. You see a linker, there's a small kind of this pin shape like this. So this become your antigen recognition domain. And here we are taking variable chain and heavy chain for CD19 from anti-CD19 antibody. Why we are picking CD19? Because CD19 is a marker of B cell. Every single damn B cell will express CD19. And if B cells are proliferating because of the mutation, you have more CD19 in your blood. So you want to delete CD19, and that's why you are taking anti-CD19 antibody SCFB. And then we made this whole construct IL-12 from, because TH1 we need, CD28 for co-stimulatory molecule, CD3 is actually TCR signaling. So TCR actually signals through CD3. So we are removing TCR, we are only taking the CD3 domain. So now we make this whole CAR T cell construct, and then we put on the T cells. So if I have a tumor, CD19 tumor, I made this construct in the lab, this one. I take out my blood through the doctor apheresis, we call it apheresis, process apheresis, you remove a lot of blood, and this blood goes to my lab, for example, and this lab, my lab will make my T cells to start expressing this CAR T cell construct through a process called lentivirus delivery. Lentivirus delivery. So this whole DNA, this is right now in the protein form, this whole DNA synthesized construct will be packed into the lentivirus, right? And the lentivirus will be now putting on the T cells, my T cells, then all T cells which is now put lentivirus will start expressing this on the surface, like this on the surface, this on the cytoplasmic tail within the cell. Now I take these T cells and I give it to doctor and say inject into me, and they inject into me. Then B cells being recognized by this, the T cell will be activated because this is in the T cell, and T cells start killing the B cells. So this is how the whole CAR T cell field is happening. And now B cell lymphoma is taken care by CAR T cell. Multiple myeloma is being taken care by the CAR T cell. Glioblastoma, which is a neuroblastoma, is being taken care by the CAR T cells, right? Autoimmune disease, which is a B cell mediated disease like lupus, is taken care by the same CAR T cell. You remove B cells, patients feel better. And by the way, these all are done, proved in the clinical trial, and they are in the market. In India, only the lymphoma product in the market, but then the US, there are multiple products in the market. And now people are trying to do is making more different CAR T cells which can target solid tumor. Solid tumor is one of the target, which is difficult nowadays to do it by the CAR T. So now this came from TH1 thing, harnessing TH1 function for adoptive CAR T cell therapy. So if you don't know the TH1 pathway, this wouldn't have been possible. So this is, yeah, the translational part of it, yeah. That's a very good question. I think she's the business lady, so I must say she's asking the cost because that's very important. We all are sitting here because of the cost, because we want to earn money, right? How many of you like to earn money? Everybody like to earn money. Everybody. But you never raise your hand. Huh? You like to earn money. You're from Andhra? Okay. Andhra people say yearn, not earn. So everybody like to earn money, not the knowledge. I need Chitvi, I need Patvi. I need both. She's saying she doesn't like to earn. Why is she always pointing you? she doesn't, you don't like to earn money? So your friend is indicating. I think she gonna have hard time in the night. She will say you did it. So yeah, so CAR T cell. Coming back to the point, I think the therapy in India is very costly. In India, is right now the two company. One is selling around 40 lakh rupees per infusion, means per injection, and and the second one is 70 lakh rupees, which is a Biocon company. In US, I think it's 4 or 5 CR. So still we are much lesser than the US cost. The idea is the whole idea, because I sit in many committees of CAR T cell in Delhi. The whole idea is to bring it to 15 lakh from 40 lakhs. And the idea is to bring it to the Ayushman Bharat scheme, so that, the people, because a lot of kids are from the poor family. I see them there, and they're having a lot of B-cell lymphoma, and the kids, the parents are crying and hell, and understand this. All emotional business is sometimes very difficult, right? So, but you have to be emotional because we are human being. But be emotional for good things, not the nonsense thing. Okay. Now, you still like to go for the TH2, or you guys are tired? You are not tired. You want me to become tired. Okay. So TH2, I'll not pay too much attention because it's a very straightforward pathway. It's more into allergy and asthma, and it's important for clearing extracellular pathogens. So in the TH2, if you really see this here, something, so the TH2 cells, it's not very clear how the initiation happens of the TH2. What TH1 is easy. IL-12 DC activation, that's what happens. But here, still kind of some confusion from where this initial IL-4 comes, which is a cytokine initiate the TH2 differentiation on the naive T cells. Some people say dendritic cells, a special kind of dendritic cells, and some people say there's a basophils. These are the granulocyte, right? And they produce initial burst of IL-4, initial burst, right? And then these TH2 cells make IL-4 by themselves, and then they perpetuate later on to make more TH2. So when I say specific kind of dendritic cells, these are the nomenclature of is IRF4 positive. These are transcription factor KLF4 positive. They make IL-4, initial IL-4. These are special dendritic cells. Then you have some of these epithelium mucus, layering epithelial cells make some of these factors, IL-25, 33, TSLP. They also contribute to ILC2 and TH2. So it's kind of hell amount of thing. Let's not go to more confusion. Let's understand that TH2 happens. Let's think that way, right? God knows who does what, but let's say you have the end product, right? And this end product is really important. Why? Because once you have these TH2 cytokines which produced by the TH2 cells, it does multiple things. It actually activates eosinophils through this IL-5, which is produced by TH2 cells. You have IL-4, which is also produced by TH2 cells, activate mast cells, M2 macrophages, and B cells class switching, and B cell IgE, what I was mentioning it, right? So these are the function of TH2 cells. So based on these two papers, I could draw this diagram just to emphasize on two things, that the initial burst of IL-4 is coming from the basophil, but activation is happening through the dendritic cells. So you have this activation is happening through the dendritic cells, class 2 signals. But during this 2 signal, this naive T cells getting activated, but the basophil is also coming in the picture, making IL-4, giving this initial IL-4, and then they become Th2, and this IL-4 now baking more and more and more, right? So only thing, initial level you need this, but later on you have more naive T cells, but those naive T cells can be driven by this IL-4 produced by Th2, right? So this is only very small amount is required in the beginning of the act initiation. So basophil is your granulocyte. You have basophil, you have eosinophils, and these are the kind of cells which is a very rare in number, mostly in the mucosal sites, right, in the lung, in the gut, and where you have the mucous lining. So basophil actually makes this IL-4, makes Th2, and then you have the dendritic cells interact with the basophil, and this interaction is called, this term called trogocytosis. So trogocytosis is a term by which, so what I'm trying to tell you that basophil itself can be antigen presenting cells in certain condition, like you can see here, basophils are expressing MHC molecule, activating it, making Th2. In some condition, the dendritic cells and basophils are interacting, and this process called trogocytosis means the MHC molecule of the dendritic cells is being transferred to basophil. And as a result, the basophil become antigen presenting cells and makes IL-4 and Th2 kind of thing. So this is the initial identification of how IL-4 actually or basophil contribute to it. Now, the second thing is the epithelial lining. Whenever we have pathogen, extracellular pathogen, they interact to the cell, this epithelial lining. The interaction of this epithelial lining with the parasite or the pathogen or extracellular pathogen tell these cells to produce some of these cytokines, IL-25, 33, TSLP, and this is through ILC2 further activate this whole pathway. So this is the whole pathway which is complex. I try to pieces out into this, and this is another way by which ILC2 is called innate lymphoid cells. It's innate lymphoid cells. Innate lymphoid cells character is that they don't require TCR activation. They don't require MHC molecule. They can be activated by the cytokine itself. So that's why these cytokine can directly activate ILC2 without T cell receptor. And this initial burst of cytokine, once they are activated by these, they make this IL-13 and they activate Th2 pathway and this Th2 like this, right? The number of ILC2 in our body is very small fraction. If you try to see them through the sort by fax machine, maybe you see some dots on them, and you have to then amplify it through some growth factor. We try to do those experiments, but they are kind of very cumbersome experiment. And this is, I'll just skip you some of this transcription factor which is required for Th2. I'll just skip it, it's kind of a bit complicated. And then based on those, you can really start thinking what are the therapeutic target of Th2. Th2 is mainly involved in the asthmatic disease. So if somebody is having allergy or asthma, like, and asthma could be very life threatening. I know older people in the colder region, they have like problem of inhaling the oxygen. So therefore they have something like, they put something like, I don't know what is the name of it, right? Inhaler or something, right? Just to, these muscles has to be smooth and so they can take the oxygen. But what you can do, you can actually, since Th2 cells are involved, so you have to, what you have to do, you have to block Th2 cells. Inhaler could be for some time, but you cannot really do inhaler all the time. So therefore you can have these target which you can see on the screen, and these target can be now used to block Th2 cells. And if you block Th2 cells, you can breathe, your muscles are relaxed. So this is the beauty of identifying all these pathways in the Th2 cells. So now you are ready, or is you are tired? I'm okay. Ready to go and have fun? क्या ही होगा यहाँ पे जाके, चाय वाय पियोगे? Ready to go and have samosa or what? Or I'm okay, so don't worry. If you're tired, I'm fine. Be frank. You are tired. I just but trying to say you are tired, which you are not. Go ahead or not. I'm saying going ahead or just stop. Please go ahead. She is very respectful. Thank you. So now why it took like almost 20 years. The first paper of TH1 and TH2 was 1986, right? And the third trilogy, the third member was discovered in 2006. It took 20 years by so-called brilliant scientists to identify the TH17 cells. Why? Right? That means there's a lot of complications. And those complications, I'll tell you if you really listen. But you have to pay more attention. You have to be like sharp. You have to stop me if you're not able to get it. Because that's the story I want to tell. Are ready to ride the bus? All with me? Pilot ready? Okay. So now what I told you, one thing is the TH1 cells. What I told you, the TH1 cells are important for clearing what? Intracellular pathogens. But when they become reactive to self-antigen, they can cause autoimmune disease. She is very brilliant, right? She is reading my lips, actually. I say auto, she says autoimmunity, right? So now, what I say that, one of the things when a patient goes to a clinic, to a doctor, and doctor, particularly in US and those developing countries, they have different tools. They can आपके brain में भी झांक सकते हैं because of those kind of instrument. So what they found that accumulation of interferon gamma producing TH1 cells in the active lesions of multiple sclerosis patient. When I say multiple sclerosis patient, these are the young individuals who are in their really 30s and they get this disease and they become paralyzed. It's a disease of the nervous system. Your T cells start chewing the nerves, right? And as a result, you have paralysis. And you have to be on the wheelchair all through your life. And that causes in the developed country a lot of burden as a GDP. Because in America or Europe, everybody has to work. It's not like that husband has to work and wife is okay, kitchen. No, both have to work. Then only you can have good life. So therefore, if one individual become paralyzed, the family is gone, the government having a lot of effort on their GDP and all those kind of stuff. So therefore, these diseases are taken very seriously in US and developed country, right? So what they have done, they found, don't read all these points, what I'm trying to say that they found in these lesions of the brain, they found more TH1 cells. So they thought the disease of multiple sclerosis, an example, it could be other disease, are TH1 associated because you have interferon gamma. It's simple, no brain, right? 1 + 1 = 2, right? But then this 1 + 1 = 2 for longer time give lot of false result and lot of false therapies, and that's how the mouse immunologists actually identify TH17 cells using one of the very important model. And this experimental model is called experimental autoimmune encephalomyelitis. It's called EAE, right? So this is a mouse model for human disease multiple sclerosis. Because आप human में direct नहीं कर सकते काम, right? Because you cannot really inject something. मेरा मन कर रहा है, चलो inject कर देते, नहीं होता है. This doesn't work, right? So you need to first make a proof of concept or POC in the animal model. Then you convince the doctor or maybe the committees that this could be potential therapy. You try in the human, in the clinical trial, phase 1, phase 2, blah, 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 and then you go for it, right? So therefore, this model become very handy. And what you have to do, you take the myelin antigen, which is expressed in the brain, right? So myelin antigen could be these different antigen, MOG oligodendrocyte, PLP or MBP, myelin basic protein. You take these things, you make emulsion, you immunize these mice subcutaneously, and in 20 days, or not 20, 10 days, these mice start seeing paralysis. So they get paralyzed. It's a very similar like human get paralyzed, right? Because of the self-reactive T cells you induce by this self-antigen. So now mice is having paralyzed, you have more T cell in the brain, and you have inflammation and all those kind of thing. And if you see the score of the paralysis, you immunize mice here, and then around 15 days, you have the highest score, which we called at 4, is a peak of the disease. That means the mice hind limbs are paralyzed, even forelimbs are paralyzed, and they are in the moribund state. We have done this experiment many, many times. And then if you just sacrifice this mouse and then you try to see the brain, you see a lot of infiltration of T cells in the brain in these mice. And these T cells are actually killing the axons and those nerves actually and all those kind of thing. And as a result, you have paralysis. So far, so good. The model is very straightforward, right? So now this brings more complications. So I told you the model. You know the cytokines. IL-12 you know. So during this time around 2000 and 2002, the half brother of IL-12, or we call it IL-12 family cytokine, called IL-23, was discovered in 2000. It's called IL-12 family cytokine. It's having some similarity with IL-12. And in 2002, the IL-23 receptor was discovered. 23 receptor was discovered. So in 2000, a new cytokine which looks like IL-12 is discovered in 2002. 2 साल बाद उसका receptor भी discover हो गया. When this discovery was happening, people thought it's maybe like IL-12. It could be the cytokine which further enhanced TH1 because of similarity of the cytokine, amino acid, and all those kind of things. So people did not really pay much attention. ठीक है, एक और नई cytokine आ गई, right? One new product in the market. इंस्टा आ गया, इंस्टा के बाद कुछ और आ रहा है, तो ठीक है, क्या ही फर्क पड़ रहा है, रियली है उसमें क्या है, राइट? बट यहां से कॉम्प्लिकेशन स्टार्ट हुआ. ये हो गया, ये तो आपको पता ही है, ये भी पता है, ये भी पता है, ये नहीं पता है. ये जब डिस्कवरी हुई तो इसमें जो एक सब यूनिट है, दिस इज ऑल्सो हेट्रोडाइमर, इसका रिसेप्टर भी हेट्रोडाइमर है. इसकी जो एक सब यूनिट है वो शेयर होती है, ठीक है, ये दोनों शेयर कर लेते हैं. और इसकी जो एक और सब यूनिट है वो स्पेसिफिक है. इसकी ये वाली स्पेसिफिक है, इसकी ये वाली स्पेसिफिक है. और इसका जो एक रिसेप्टर है वो भी शेयर्ड होता है. बीटा 1 इज हियर, बीटा 1 इज हियर. ये बीटा 1 इससे पेयर करता है बीटा 2 से, ये बीटा 1 IL-23 रिसेप्टर अल्फा से पेयर करता है. तो ये जो कॉम्प्लेक्स हो गया, ये IL-12 रिसेप्टर हो गया, ये 23 रिसेप्टर हो गया, ठीक है. नाउ आप चाहकर भी इसको अलग नहीं कर सकते. आप कहोगे यार ये तो IL-12 की वजह से हो रहा है कि IL-23 की वजह से हो रहा है, क्योंकि ये भी शेयर्ड हो रहा है, ये भी शेयर्ड हो रहा है. तो एंड में बोलोगे भाड़ में जाओ यार, व्हाई द हेल आई हैड टू डू दिस, राइट? जाके चाय पीते हैं, क्या करना है, राइट? यही होता है ना. एंड में जब दिमाग का दही होता है तो क्या करते हो? यही करते हो, टीवी देखोगे, चाय पियोगे, चलो यार चलो बाइक निकालो, राइट? एट इस स्टेज, राइट? फिर घूम घाम के आओगे, फिर भी तुम में जिसमें कीड़ा होगा, यार करना तो है इसको सॉल्व, कैसे करूं, ये दिमाग में घूम रही है पिक्चर, सॉल्व करना है, पजल है, इट्स अ पजल. तो इस पजल को सॉल्व करने के लिए इतने तरह के जो नॉकआउट माइस यूज करे गए. आई टेल यू द स्टोरी. आप ऊपर हेडिंग पढ़िए, TH1 सेल्स एंड इस एसोसिएटेड मॉलिक्यूल, ठीक है. ये सारे मॉलिक्यूल मैंने आपको TH1 में बताए थे, सारे देखो. ये TH1 का था, ये TH1 का था, ये TH1 का था, ये भी तुम्हारा, ये साइटोकाइन है तुम्हारी IL-12, ये भी IL-12 ही हो गया. ये IL-12 रिसेप्टर का है, रिसेप्टर का 1 यूनिट. ये इनको हमने माइकोबैक्टीरियम में टेस्ट किया था, है ना. सब माइस मर गए थे, टीवी ग्रो हो गई थी. अब इसको हम दूसरे मॉडल में यूज कर रहे हैं, EAE मॉडल में, ठीक है. वो TH1 वाला मॉडल था, ये भी लोगों ने सोचा TH1 इसमें भी टेस्ट करते हैं. तो इन सबको इम्यूनाइज कर दिया माइलिन एंटीजन से, और फिर देखा कि यार अगर सच में TH1 इंपॉर्टेंट है इस बीमारी के लिए, जो कि लीजन्स में भरा पड़ा है, डॉक्टर कह रहे हैं ना लीजन में आइफन गामा है, तो अगर आइफन गामा रियली इंपॉर्टेंट है तो, और आइफन गामा वाला पाथवे इंपॉर्टेंट है TH1, तो इनमें बीमारी होगी नहीं होगी अभी मैं बताता हूं, ठीक है। आप देखो यहाँ पे पैरालिसिस हो गई, अगर आइफन गामा इंपॉर्टेंट होता तो पैरालिसिस नहीं होती, है ना, बिकॉज पॉजिटिवली कोरिलेट हो रहा था ना पेशेंट में, आइफन गामा है बीमारी है। अब हमने क्या किया, माइस में आइफन गामा हटा दिया तो बीमारी हट जानी चाहिए, यहाँ तो उल्टा और ज्यादा बीमारी हो गई। तो इससे क्या कंक्लूजन निकला, TH1 इंपॉर्टेंट नहीं है। यहां भी वही हुआ, TH1 इंपॉर्टेंट नहीं है। तो पेशेंट के डेटा को हम यहाँ पे माइस में बोल रहे हैं कि वो जो आप देख रहे हो, दैट कुड बी एन आर्टिफैक्ट, इट मे नॉट बी रिस्पॉन्सिबल फॉर योर डिजीज EAE। सारे नो TH1, सब में पैरालिसिस हो गई, ठीक है। हाँ और कौन कह रहा था नो पैरालिसिस? दैट मीन्स यार यू गाइस आर वेरी स्मार्ट। मैं तो सोच रहा बेवकूफ बना के आगे बढ़ जाते हैं, अब दो पकड़ लिया तुमने, दो लोगों ने। नो पैरालिसिस कहाँ लिखी है? ये यहाँ पे पीछे यहाँ पे लिखा है। तो अब इसका मैं क्या करूं? अब ये और कंफ्यूजन हो गया। हाँ, तो अगर आप ये इसको छुपा देते हैं थोड़ी देर के लिए तो सब स्टोरी बढ़िया चल रही है, ठीक है, एकदम परफेक्ट फेरी टेल हो रहा है, बढ़िया। स्विट्जरलैंड की ट्रेन में बैठे हुए हैं, जा रहे हैं, राइट। I was 5 days ago, I was in Switzerland, so I came for this meeting. So then this happened, then it looks like यार ये बड़ा confusion सा हो रहा है कि इसमें TH1 भी नहीं है, इसमें TH1 भी नहीं है, बीमारी भी है, इसमें TH1 भी नहीं है, फिर भी बीमारी नहीं है, either way. So ये एक्सपेरिमेंट को सॉल्व करने के लिए now this experiment has been done which got published in Nature in 2003 that actually dissect everything. आपका जो मेजर कंफ्यूजन हो रहा था वो यही हो रहा है, ये यहाँ पे इसी में हो रहा है, राइट। तो इसको डाइसेक्ट करने के लिए 3 नॉकआउट माइस उन्होंने लिए, एक लिया P35, एक लिया P40, एक लिया P19, ठीक है। ये जो दोनों हैं, ये दोनों होंगे तो ये बनेगा, ये दोनों होंगे तो ये बनेगा। अगर ये माइस ये इसमें कुछ नहीं हो रहा है तो इसकी वजह से भी हो सकता है, इसकी वजह से भी हो सकता है ना। अगर इस चीज में कुछ चीज हो रही, कोई फिनोटाइप आ रहा है, तो या तो इसकी वजह से है या इसकी वजह से है, राइट। तो अब हमने इसको कर दिया इम्यूनाइज, पैरालिसिस हो गई, TH1 नहीं है फिर भी पैरालिसिस है, यहाँ पे TH1 नहीं है, यहाँ पे P40 TH1 नहीं है, नो पैरालिसिस, TH1 नहीं है, और यहाँ पे TH17 भी नहीं है, ठीक है। Because TH17 भी नाउ आफ्टर बिफोर दिस डिस्कवरी TH17 स्टार्टेड कमिंग टू कि यार ये डिजीज में काम आता है। तो यहाँ पे TH1 भी नहीं है और यहाँ पे TH17 भी नहीं है। That means P40 is important for both TH1 and TH17. और यहाँ पे अगर P19 हटा देते हैं, ठीक है, तो बीमारी नहीं है, TH1 है, TH17 नहीं है। तो इससे क्या कंक्लूजन निकल रहा है? इससे ये कंक्लूजन निकल रहा है कि जो IL-23 है वो बीमारी के लिए इंपॉर्टेंट है, IL-12 नहीं है, क्योंकि यहाँ पे तो IL-12 चला गया फिर भी हो रही है। तो IL-23 जो पाथवे है, चाहे इसको डिलीट कर दो चाहे इसको डिलीट कर दो, बीमारी IL-23 की वजह से हो रही है और बीमारी TH17 की वजह से हो रही है। यहाँ पे TH1 है, इसमें नो बीमारी नहीं है। तो जहां जहां IL-23 है और जहां जहां TH17 है वहां वहां बीमारी हो रही है। दैट वाज द कंक्लूजन ऑफ दिस पेपर। आई होप यू अंडरस्टैंड दिस, राइट। अगर आप ये माउस ये वाला नहीं बनाते, ये वाला नॉकआउट, तो आप इस पजल को सॉल्व नहीं कर पा रहे थे। आप हमेशा कहते हैं कंफ्यूजन हो रहा है, इसमें बीमारी, इसमें पैरालिसिस हो जाती है, इसमें नहीं होती, दोनों IL-12 के हैं, तो दोनों में सेम रिजल्ट क्यों नहीं आ रहा है? वो इसलिए नहीं आ रहा है क्योंकि ये शेयर हो रहा है यूनिट, राइट। सेम थिंग इन्होंने करा रिसेप्टर में भी किया था, सेम रिजल्ट इन द रिसेप्टर बिकॉज ऑफ द शेयरिंग। अब इसी टाइम पे क्या हो रहा था कि ये एक्सपेरिमेंट हो गया, 2001 में हो गया था, राइट। लोगों ने सोचा कि यार TH17 इज द मेन कल्प्रिट, अब TH17 का डिफरेंशिएशन फैक्टर निकालो, जैसे TH1 का IL-12 है, ठीक है, TH2 का IL-4 है, तो लोगों ने कहा अगर TH17 इतना कल्प्रिट है बीमारी के लिए, तो इसके पाथवे क्या है? तो एक कैम्ब्रिज में एक्सपेरिमेंट हो रहा था, एंड ड्यूरिंग दिस एक्सपेरिमेंट व्हाट दे वर ट्राइंग टू डू, दे वर टेकिंग 3 डिफरेंट सेल टाइप्स फ्रॉम माउस। एक सेल टाइप्स वो ले रहे थे, नाइफ टी सेल्स निकाला उन्होंने, सॉर्ट किया, एक डेंड्राइटिक सेल्स निकाला, एक उन्होंने रेगुलेटरी टी सेल्स होता है जिसका ट्रांसक्रिप्शन फैक्टर होता है FOXP3, ठीक है, वो तीनों को जो को कल्चर किया साथ में, वेल में डाला तीनों को, एंड देन दे ट्राई टू सी कि TH17 डिफरेंशिएशन होता है कि नहीं, तो इसमें नो डिफरेंशिएशन। जैसे ही उन्होंने इस कंडीशन के साथ LPS डाल दिया, LPS, तो TH17 डिफरेंशिएशन हो गया। तो इसमें तुम्हारे 4 कंपोनेंट, 3 कंपोनेंट हो गए, ये तो बन ही जाता है, नाइफ टी सेल तो बना ही दे रहा है, तो तुम्हारे इसमें 3 कंपोनेंट हो गए, 1, 2 और 3। यहाँ पे 2 कंपोनेंट है तो नहीं हो रहा है, यहाँ पे जैसे ही तीसरा कंपोनेंट LPS डाला तो हो गया, ठीक है। तो फिर लोगों ने क्या सोचा कि LPS होता है वो तो डेंड्राइटिक सेल पे ही काम करता है मेनली, उसका जो TLR4 रिसेप्टर होता है वो डेंड्राइटिक सेल पे ही ज्यादा होता है, इन वाली सेल्स पर नहीं होता है। तो लोगों ने सोचा, मतलब इनके जो ऑथर्स ने, कि LPS से जो डेंड्राइटिक सेल्स एक्टिवेट हो रही है, ठीक है, और डेंड्राइटिक सेल एक्टिवेट हो रही है तो वो बना रही है IL-6। तो इन्होंने क्या किया कि डेंड्राइटिक सेल्स और LPS को रिप्लेस कर दिया IL-6 से, और Trex को रिप्लेस कर दिया TGF-beta से। TGF-beta एक इनहिबिटरी साइटोकाइन है, TGF-beta is inhibitory cytokine which is produced by the regulatory T cells। सो रेगुलेटरी टी सेल से भी कोई सिग्नल मिल रहा था और डेंड्राइटिक सेल और LPS से भी एक सिग्नल मिल रहा था, तो उन्होंने दोनों सिग्नल को, इट्स अ बिग एक्सपेरिमेंट, दे हैव डन मल्टीपल परम्यूटेशन कॉम्बिनेशन ऑफ डिफरेंट साइटोकाइन, द वे आई एम सेइंग इज नॉट लाइक दैट, but they come to a conclusion that IL-6 and TGF-beta 2 together can make naive T cells to become TH17, and that's what they publish in Immunity। इसी टाइम पे एक और ग्रुप काम कर रहा था, where I was working in Harvard, so they have created a mouse, it's called TGF-beta transgenic mouse। TGF-beta क्या है? इनहिबिटरी साइटोकाइन है। तो इस माउस में क्या होता है कि TGF-beta भर भर के निकलता है, बिकॉज ट्रांसजीन लगाया आपने, सो जैसे ही टी सेल एक्टिव होता है TGF-beta निकलेगा। तो एक्सपेक्टेशन हो रही थी कि जब हम इस माउस को इम्यूनाइज करते हैं वो जो माइलिन एंटीजन से, तो TGF-beta बनेगा, एक्टिवेट होंगी टी सेल, इम्यूनाइजेशन में टी सेल एक्टिवेट होंगी, TGF-beta बनेगा, तो क्या होगा, हमें ये रिजल्ट चाहिए था। हम सोच रहे थे यार कि उसमें तो TGF-beta एज ए सप्रेसिव साइटोकाइन, बीमारी होनी नहीं है, एक्सपेक्टेड रिजल्ट है, दैट्स इट, ये आना चाहिए, आ रहा था ये, हर बार इम्यूनाइज करो ये रिजल्ट आ रहा है। एवरी टाइम वी गो एंड डिस्कस इन द लैब मीटिंग PI से, यार या क्या बकवास है यार, घर सही से नहीं कर रहे एक्सपेरिमेंट, राइट, ये तो उल्टा ही हो रहा है, वो फेक दे रहा है रिजल्ट, गेट लॉस्ट, डू इट अगेन। देन व्हाट हैपेंस, and this is we publish again in Nature, parallelly another postdoc in the lab was working on another mouse, ठीक है। I told you that FOXP3 positive, FOXP3 is a regulatory T cell transcription factor, so उसी टाइम पे FOXP3 की डिस्कवरी हुई थी। There are 2 very good scientists, one in Japan, Simon Sakaguchi has identified this transcription factor, now a lot of work is happening on this। so ये नया ट्रांसक्रिप्शन फैक्टर था, तो लैब हैज जनरेटेड अ माउस वेयर वी लिंक्ड दिस ट्रांसक्रिप्शन फैक्टर विद द GFP, एंड द आइडिया, KI मतलब नॉक इन, GFP को नॉक इन किया है, नॉक इन, राइट, नॉक आउट, नॉक इन, नॉक आउट मीन्स रिमूव, नॉक इन माने डाला, इंसर्ट किया। Now, the idea was to identify all the cells in the mouse, in vivo, inside the mouse, should become green so that we can see them. फैक्स के थ्रू देख लो, कन्फोकल के थ्रू देख लो, नीली नीली हरी हरी चमकेंगी, राइट. एंड देन वी कैन टेक देम आउट, वी कैन सॉर्ट देम. इनका मार्कर तो कुछ था नहीं, सरफेस मार्कर. वी कैन सॉर्ट देम एंड से क्या प्रोड्यूस करते हैं, क्या ट्रांसक्रिप्शन है, ब्ला ब्ला, ऑल दिस काइंड ऑफ डिस्क्रिप्टिव वर्क. So using this mouse, the thing was, can we convert non-FOXP3 cells, which is majority in nature? 90% cells of CD4 cells are this. Okay, this. 10% cells of CD4 are this. Okay? But these 10% are so important that if you remove these cells, 10% from the mouse or human, you die. It's so important. So people has created a mouse, it's called Scurfy mouse, in which there is a naturally occurring mutation in the FOXP3 gene. That means the FOXP3 is not expressed, and as a result the Treg cells are not generated, and mice die in 4 weeks. In the humans, the FOXP3 natural mutation has been identified in the human. It's called IPEX syndrome. It's a big name, IPEX syndrome. And these individuals also die in the very young because of the multiple organ failure. And why multiple organ failure happens? Because if you remove these cells, these non-Treg cells become crazy, very activated. They go to brain, they go to liver, they go to kidney, they go to every single vital organ, and your vital organ is like dying. Same thing happens in the mouse, same thing happens in the human. So what I'm trying to say that this 10% sometimes जो छोटी चीज होती है ना is very important. वो कहते हैं ना सुई का काम तलवार नहीं कर सकती, right? This is exactly what I'm trying to say. Sometimes small thing is so important for you in your life that you cannot ignore, right? So the idea was, if these cells are so important, it's a question of life and death both in mouse and human. Can we identify the factors by which we can increase these cells in the autoimmune disease setting? Autoimmune disease setting में हमें इन cells को बढ़ाना है, क्योंकि autoimmune को कंट्रोल करोगे तो ये वाली इसको कंट्रोल करेगी और बीमारी इसी से हो रही है. तो उसको करने के लिए एक साइटोकाइन है जिसको बोलते हैं TGF बीटा, बताया था. तो हम क्या करते थे, we used to sort these cells out, ग्रीन वाली हटा देते थे, we used to sort them out, activate them with those 2 signal and put TGF beta, they become green. They all become green. Now what we wanted to identify, what are the other cytokine in combination of TGF beta, make them more green or less green. That was our idea. So we did this experiment. We took the panels of cytokines. Here you see TGF beta alone, you have 100% cells. Non-FOXP3 becoming FOXP3, non-GFP becoming GFP. That is your control. And then you are adding multiple cytokine, IL-1, 2, 6, 7, blah, blah, all this kind of thing. And you see some cytokine decreasing this, and one of the cytokine which is decreasing the highest from 100% to become around 30% is your IL-6, right? And when you collected the supernatant from all these cultures, and then you do IL-17, the IL-6 and TGF beta, where you suppress FOXP3, you have more IL-17, right? And that's how the paper came out in Nature, where we show this, that naive T cells, TGF beta and IL-6, the 3 paper came. One was immunity, I told you, Tregs or co-culture, and they also say TGF beta and IL-6. The other one is this Nature, and other one and the Nature, that naive T cells activate 2 cytokines, put on this start differentiation. And this differentiation make these cells Th17, they start all these receptor, and then this IL-23, which was other mouse I showed you. Actually, IL-23 make these cells more pathogenic and more survive. That's why the result in the IL-23 knockout, they are resistant, because this is also very crucial, as crucial as these. This is for initiation, this is for maintenance of these cells. And this is what we published, this paper. So this was the story of triology of Th17 cells, and it took 20 years to identify. And now this is something I just skip. I think you have heard a lot about this. ये जहाँ जोड़ दो हटाओ MCO kind of stuff, right? Do you want to hear it? I think you are bored already. Acetyl co निकलता है, ये mitochondria stuff, right? I just try to put something just to make me feel I am teaching epigenetics, है ना? Just to fool you all. ये हटा देते हैं. इसमें लास्ट में ये है, हाँ, ये फिर से आ गए इनके पास. स्टार्ट इन्होंने किया था, भगवान ने. भगवान स्टार्ट करते हैं, भगवान ही अंत करेंगे, है ना? क्रिएटर एंड डिस्ट्रॉयर आर शिवा. वेरी क्लियर. So and that means God is creating us and God is destroying us. All of you believe in God. I do also believe in God, right? All you? पक्का? रात में घर जाके तो नहीं आ रहे क्या गॉड है? हॉस्टल में जाके डिफरेंट अवतार है, अब छोड़ो गॉड को, है ना? यहाँ पे यस, आई एम वेरी civilized person, I believe in God, right? So ये इन लोगों के पास हम फिर जाते हैं, तो इन्होंने हमें क्या बताया? इन्होंने ये बताया कि आप ये करोगे तो ये होगा, ये करोगे तो ये होगा, ऑटोइम्यूनिटी भी हो जाएगी, और या फिर ये पाथवे है. जो इनसे जो एक चीज छूट गई थी, जिससे कई लोगों ने अपना करियर बना लिया, अगर ये उसी टाइम पे डिस्कवर कर देते तो कई लोगों का करियर तबाह हो जाता, जो 2006 के बाद बना है, और तब से अभी तक उस पर राइट कर रहे हैं. अरे उसमें मैं भी एक हूँ, मैंने भी अपना करियर इसी पे बना दिया. सो द क्वेश्चन इज दैट यू हैव two sets of cyto cells, Th1 and Th2. One takes care of the intercellular pathway, forget autoimmunity for time being, and one is extracellular. तीसरी चीज जो ये दो सेल टाइप्स नहीं कर पा रहे थे, वो था एक फंगल पैथोजन. फंगस होता है ना, नाखूनों में ग्रो करता है, बालों में ग्रो करता है. एंटी डेंड्रफ शैम्पू आप यूज करते हो बिकॉज ऑफ फंगस. हेड एंड शोल्डर में कुछ नहीं है, एंटी फंगल चीजें हैं, आप घर में बना सकते हो, वो अमेरिका से मंगाते हो. इट्स ऑल फंगस. इफ यू हैव मोर फंगस, you have this white flakes, and you should not wear black shirt like I am. So every time I have to do. वो होता ना, वो हेड एंड शोल्डर के एडवर्टाइजमेंट में वो एक बंदा होता है काला कोट, उसके बगल में आके ऐसे ऐसे कोई कर देता है, जस्ट टू मेक श्योर ये डैंड्रफ. तो फंगल है, ठीक है, उंगलियों में लग जाता है, टंग पे आ जाता है. तो वो फंगस को Th1 भी क्लियर नहीं कर पाता और Th2 भी क्लियर नहीं कर पाता है, ठीक है. तो वो क्या का होता है, उसके लिए Th17 है, हर मर्ज की दवा, इम्यूनोलॉजी, राइट. सो उसमें क्या होता है कि यू हैव एक्स्ट्रासेलुलर फंगल पैथोजेंस, and how it actually identified in the Europe or European country, there are families in which there's a natural mutation of the Th17 pathway, like STAT3. I told you STAT4 Th1, STAT3 for Th17, STAT6 for Th2, right? STAT4 Th1, STAT6 Th2, STAT3 for Th17. तो STAT3 में लोगों ने देखा कि म्यूटेशन है ह्यूमंस में, और वो ह्यूमंस क्या होते हैं, दे आर वेरी ससेप्टेबल टू फंगल इन्फेक्शन, and at the same time they don't have Th17 response. So people have identified a kind of correlation of Th17 to the fungal infection. If you have more Th17, it can control your fungus. If you are less Th17, you cannot control your fungus, because these two guys can't take care of it. So therefore the triology of these T cells can take care every single kind of antigen, and that's why the Th17 cells are absolutely important. This is my lab. This was the picture perhaps last year. Every year we take a new picture just to see how much older we are growing, right? And we are growing very fast older. So try to do what you can do. Time is not stopping for you. So you try to do whatever you like to do. and sometimes you do wrong thing, then you say sorry, but keep doing it. If you don't do it, you are dead, right? So these are all brilliant. You see their face, they're kind of very brightened, smiley, handsome. Everybody is looking so handsome. Look at this guy, that kind of Tushan he has, like with me, right? And all these are working very, very hard. Some of them actually left for the postdoc to US or Singapore or maybe European countries. Some are doing still PhD and they're trying to again go abroad or stay here, whatever they like to do. So that is my lab, and this is all stuff. That's it. So if you have any question, I'll be happy to answer. And you have to delete my slides. आप मेरी slide delete कर देना. I think he did not get it. Yeah. Any questions? Any suggestion? Any advice? I hope you got everything very clear. So try to take immunology as your PhD. You have immunology course here, third semester. Same thing. All India में वही होता है, third semester. इम्यूनोलॉजी को थर्ड सेमेस्टर में पढ़ा देते हैं. क्यूबे सेकंड, क्यूबे हाँ सेकंड सेमेस्टर. क्यूबे पढ़ते हो? क्यूबे और कौन सी बुक पढ़ते हो? राइट्स एंड राइट्स पता ही नहीं, पढ़ा ही नहीं है मैंने. क्या क्यूबे पढ़ते हो? हाँ ऑलरेडी डन. समझ में आया था? इम्यूनोलॉजी वो थोड़ा, मैं भी जब आई वाज डूइंग एमएससी आई वाज लाइक आल्सो लाइक दिस. समझ भी नहीं आती. इट्स लाइक स्टोरी. किसी को somebody has to tell you the story, and when you get into it you just love it. You say यार यही है, असली चीज यही है. बायोकेमिस्ट्री में तो वही होता है, बायोमोलिक्यूल वगैरह. क्या था? क्रैप साइकिल, नॉट क्रैप्स, क्रैप साइकिल ना. Less autoimmunity. It's called trained immunity. It's I was very recently, I think 5 days ago, I was in Vienna. तो वहाँ पे conference थी immunology की, one of the biggest conference, and they have presented a data. It's very mesmerizing, and I think so. What they have done, they did an experiment in humans, and what they have done, they have taken मिट्टी जो हम लोग खेलते हैं बचपन में. आई डोंट नो तुम सब खेले होगे, मैं तो बहुत खेला हूँ, है ना. खाते भी थे, राइट. पिटाई भी लगती थी, मिट्टी खा ली, राइट. सो you take this and they feed the young people with this, let them play, and you have other group of the people who are very clean, they will not exposed to this. And then they did an experiment of allergy, and they give some allergen to these two group. One played with this dust and all, like, you know, and one was very clean. And they found you have more allergic response to the people who were very clean, right? So this is why the world is going back to what we were taught in our old ancient whatever India you call it, कि आपको expose करना चाहिए अपने आप को pathogen से, because the dust has a lot of pathogen, bacteria and all those kind of stuff, and this will make your immune response not too hyper reactive. Whenever you get a virus or something, it will have reaction, but other things will make it suppress. So you will not have more allergy, you have less autoimmune disease and all those kind of things. It's like same thing. If you really, I can give you bad example. I don't remembering the good example, but don't take my bad example in a bad way. Let's say you actually go for the first time you are drinking alcohol, very first time. And somebody who drinks alcohol on regular basis, not daily basis, they become bit resistant. उनको ठीक है, क्या ही फर्क ही नहीं पड़ रहा. पर आप पहली बार जब जाते हो तो आपको बहुत फर्क पड़ता है. आपको लग रहा है दुनिया गोल है, मैं घूम रहा हूँ या दुनिया घूम रही है, right? So this is why if you are unexposed to something and you get suddenly exposed, you have this reaction. And this is what happened in the Europe and America during COVID-19, because their immune system was so clean. And suddenly the virus comes in and their immune system is reacting like a havoc and making more cytokines, and they are dying more than us, right? We died because of some other reason, Delta में जो हुआ था, oxygen नहीं मिली, that's our problem of healthcare system. It's not because of us. इस healthcare से oxygen ही नहीं मिलेगी तो मरना ही है वो तो, right? So we did not die because of the cytokine release syndrome. We die because of the congestion, oxygen नहीं, अगर तुम oxygen लगा देते तो ठीक हो जाते, कोई problem नहीं. So my point is, if your immune system is not really exposed to some of these cues, you will react very quickly and very fast. And that's why the question to you, that the answer to you is कि आपको autoimmunity इसलिए नहीं होती क्योंकि यार exposed हो काफी. I hope I answered you. Any questions? Anybody?
Sir, कार्डिफेल में आपने बताया था कि we use lentivirus vector, but in lentivirus vector there are also some lentiviral parts also that get flown through. So
yeah,
it will make autoimmunity also.
So lentivirus vector, what we are using is taking the some part to make like virus kind of particle, but we are removing all viral antigen, which otherwise can cause some of the thing, right? So we are just using the part which is not really important for causing any illness inside. If you just, I give you that lentivirus, it will not cause any disease. So they are kind of inactivated in some way.
Can you use non-viral vectors?
That's a very good question. I think you are very smart. I think non-viral vectors people are trying. There are LEFs, there are CAR T cell therapy which was developed in Japan and finally rolled out in Thailand. They are on the non-viral vector based, but don't tell me nanoparticle is too far. लोग कहते हैं nanoparticle से कर लेते हैं, नहीं होता. Any more questions? All fine? That means you got it. All good? You got it? Anybody? Calcutta? Fine. Allahabad? All fine. All the best. So try to become member of Indian Immunology Society. You can find a form on I don't know website works or not, but I do have form. इंडिया में चलता नहीं ना, वो लाइट चली जाती है, राइट? सो so I think I will share the form with the UPI, which is more important than the form, UPI, right? And so this guy Santosh, I'll tell, and he can share with you guys, so you become member. And this year the conference is happening in Prayagraj, by the way, in November. Okay, so Prayagraj, there is a conference, and next year I think is Kashmir. So Kashmir, we want to make a conference in the Kashmir. You are from Kashmir? I thought I said Kashmir. The Kashmir I want to make sure, because Kashmir is very important part of India. I want to make sure after Pahalgam attack, we want to do more programs in Kashmir just to emphasize that we love Kashmir and that's our Kashmir, right? So that's what we are doing next year in Kashmir. And yeah, 2027, I'm organizing a big conference in New Delhi in October. So these things you will get all update when you become member. And this conference is FIMSA conference, is called Federation of Oceania Asia countries, all this Australia, Japan, Singapore, China, all this kind of side ki country hai, or Iran hai idhar, all this kind of. So these countries, we are organizing conference where people will come, or delegates will come from all over the world, including US. And that could be big conference in Bharat Mandapam, perhaps. You know Bharat Mandapam? Yes. Yeah, there was G20. Pragati Maidan was used to be. Now, yeah, G20 was done as an inauguration. So that will be a good venue for this to showcase, yes, we can do. I think all of us have to showcase India somewhere in some capacity, just to tell that we are ready and don't stop us. Put 100% tariff, but we will survive. Okay. All the best. Thank you very much.